Hello and welcome to the Stephen Academy Online Video on Demand AP Biology Update. In this series, we will be examining some of the changes that have occurred to the AP Biology curriculum and the content of the material that will be assessed on the AP Biology test. I'm your host, Dr. William Tokes. In the AP Biology exam, there are a number of things you should be aware of. First off is the overall format of the exam. As it is currently envisioned, the AP Biology exam has a structure which requires 3 hours of total time for the test taker. During this time, you'll be divided into answering short answers and long answers. For the short section, which will comprise 50% of your grade and will require 50% of the allotted time, so 90 minutes. In this time frame, you will have 63 multiple choice questions, MCQs. These MCQs are 4 answer choices from which you are to select. In addition, the College Board has removed any penalty for guessing. So if you get to the end of your test and you find that there are still multiple choice questions that you have not answered and you're out of time, it is still in your interest to go ahead and guess an answer. Even if you cannot eliminate any of the answer choices, still go ahead and guess, okay? Because there's a chance you could guess right. In addition to these 63 multiple choice questions, there will be 6 grid-in questions, which require you to perform a mathematical computation and then input your answer on the grid. These questions are a little trickier and take a little bit more thought than the multiple choice questions. As far as your timing goes, I would recommend spending a minute about on each of these 63 multiple choice questions and then spending slightly longer on the grid-in questions, 3 or 4 minutes perhaps, so as to give you time to go over your answers at the end. If you do find with the multiple choice questions that you've been staring at the same question for 5 minutes straight, it's time to go ahead, put in a guess, and move on to the next question, okay? Because you don't want to get to the end of your time and find that there are questions you have not yet considered. In addition to the short answers, you will also be required to answer longer form questions. This will also provide 50% of the grade of the test, as well as require 50% of the time, so another 90 minutes. During this time, there will be 8 questions that you have to answer. 6 of those will be short paragraph form, short, long responses, if you will. They should comprise several sentences and should do a thorough job of explaining whatever concept is being inquired about. These short answer questions are accompanied by 1 long essay form of question, which will ask you to delve into detail on a certain topic. This one should require several paragraphs, and you would be advised to follow a standard 5-paragraph essay format. Finally, you will have 1 long-form data-based question, which may be based on a laboratory where they give you a description of the experiment that is being performed or of the situation that is being observed and ask you to make determinations about the overall quality of the scientific work. The main goal that you should stress whenever you're answering long and complex questions like this, as you're reading the question, go through and underline the specific questions that they want you to answer in your essay. It's easy to get distracted and in your essay talk about things that do not specifically have to do with the actual specific question that they want you to answer, that they are going through and giving you points based on your analysis of. So just keep in mind as you answer the long questions, what is the question that I'm supposed to be answering here? In general, your preparation for the test should include also a good night's sleep and a full dinner and breakfast before you take the test. You can't take the test well if you're hungry and falling asleep, of course. Now, let's take some time and let's consider some of the specific changes to the curriculum that have been made in recent updates. One major update to the AP Biology curriculum concerns how the student may be asked to deal with data in support of their understanding of the flow of energy through an ecosystem. Properly thought of, the role of photosynthesis and respiration in the flow of energy through a system can be demonstrated with data. A typical thing you may find is that you are presented with some form of data about a system indicating that some substance is being produced or consumed by some biological or chemical reaction. In these instances, it is crucial that you recall the role played by photosynthesis and respiration and how the energy that is produced is transferred through the rest of the system. First of all, in the classically taught form of energy flow, all energy which enters the system comes from the sun. It may be thought of here in this simplified diagram that the energy from the sunlight is utilized by photosynthetic organisms, which the most efficient form of which utilize the molecule chlorophyll contained in organelles called chloroplasts. I want to stress here that there are many different forms of photosynthesis which are practiced by a wide variety of organisms which might or might not be recognizable as plants. Some of them rely on sulfide, for example, rather than carbon dioxide and water. So this is a very broad collection of potential reactions. Don't think that there's only one form of photosynthesis and everybody does that, okay? But for the purposes of the AP Biology exam, generally speaking, energy enters the system through sunlight acting on chlorophyll, which allows producers to convert carbon dioxide and water into oxygen and carbohydrates. However, it should be pointed out that the actual process is substantially more complex than merely hooking things together and building a little model, so to speak. In actual fact, what's occurring is the water acts as a hydrogen donor, which produces a gradient of hydrogen concentration within the chloroplasts. Part of the reason why cells use chloroplasts and mitochondria to perform their activities of cellular respiration and photosynthesis is that by dedicating the resources and architecture of these organelles to these chemical processes, they can be performed with substantially greater efficiency. In both cases, there is an outer membrane to the organelle as well as an inner membrane. The inner membrane may be thought of as serving to form a barrier between an area of high proton concentration and an area of low proton concentration. What is essentially happening in both photosynthesis and cellular respiration is a source of energy is used to maintain or increase that proton gradient. As the protons seek to cross the inner barrier or the inner membrane of either the mitochondria or the chloroplast, that movement is used to run microcellular machinery to perform tasks such as the replenishment of ATP. Now, in the case of carbohydrate respiration or metabolism, there are two options which you are likely to encounter on the AP Biology exam. A carbohydrate when produced through a photosynthetic process, may be broken down through either aerobic or anaerobic means. It is likely that you will be presented with some experimental situation and asked to determine which form of carbohydrate metabolism is occurring. In the case of an aerobic metabolism of a carbohydrate, oxygen is available in the system to accept the protons which are donated through the process of breaking down the carbohydrate. All right? So, as a result, water is produced as well as carbon dioxide. This is a very efficient process when conducted in a mitochondria due to the reasons I mentioned previously of the advantages of creating a proton gradient. It may be considered that in full metabolic breakdown of this carbohydrate, all 12 of those protons are removed and placed on one side of the inner mitochondrial membrane. And they form thus part of the gradient, which is used to turn various pieces of microcellular machinery. That's very efficient, and it enables the cell to produce a large amount of ATP from the energy that is released. By using oxygen as an acceptor for all of that hydrogen, the process can be conducted very efficiently especially if you have a mitochondria to do this in. However, if oxygen is not available, or if an organism is not comfortable working with oxygen, which is common because oxygen is rather deadly, they may proceed to utilize the carbohydrate metabolically through the process of fermentation. Fermentation is similar to respiration in that it does produce carbon dioxide and ATP. However, the amount of ATP that is produced is substantially less, which is why it is generally only pursued by microcellular organisms who have a relatively low energy budget. In the case of fermentation, however, no water will be produced as occurs in aerobic respiration. Instead, ethanol or alcohol will be the product. Now, in a data-based question, you may be provided with data regarding the concentrations of any of these molecular substances that are known to be part of the motion of energy through this system. Your wisest course of action is to recall which processes produce and consume which molecular substances as a consequence of their action. For example, if you are provided with data that suggests that an organism is producing carbon dioxide, that alone is insufficient to determine whether fermentation or respiration is occurring, and you would not be able to answer one way or the other. It would be appropriate in that case to ask whether ethanol or water was being produced in addition. Generally speaking, the amount of these substances which are produced through these chemical actions can be quantitatively measured, and you may see some evidence such as this provided for you to draw conclusions from. In that case, you may safely assume that the consumption rates and production rates you are seeing are the result of one of these processes, and make your determination an argument based on your understanding of these chemical patterns. an in-depth examination of the enzymatic steps which are necessary for each of these reactions is advisable as well as entertaining. However, remember that the purpose at each stage is the production of ATP, and make particular sure that you are fully versed in what that substance does within any living organism. Generally speaking, it is the source of all energy in the biological systems which rely on a fuel to function, such as the transcription of DNA, the translation of new proteins, the packaging of materials in the Golgi apparatus, the movement of the cell, as well as the many chemical processes which it performs. Without ATP, none of these would be possible. Okay. Another update to the AP Biology curriculum concerns the treatment of the immune response in organisms. In previous versions of the AP Biology curriculum, it was thought that the lower forms of life, such as plants and invertebrates, did not possess specific immune responses. However, time and diligent research has shown that immune responses are a common part of most life that exists today on Earth. Because the original forms of life which existed on Earth did so in an environment where other organisms were not adapted to invade or parasitize them, it is reasonable to suspect that early life forms lacked the complex immune responses which are evident today. However, given that a multiplicity of organisms have evolved to prey upon the resources which organisms have in today's ecosystem of Earth, it is necessary for any organism to maintain a complex suite of defense mechanisms. In the case of knowledge you will require for AP Biology testing, you should recall that multicellular organisms have non-specific immune defenses of many different types and effects, and you should be able to cite examples of these. In a plant, for example, a tree, you could refer to the passive defenses of a hard bark on the outer surface, which prevents organisms from easily penetrating it. You might also refer to the chemicals which are in the sap of the tree, which may be poisonous to other organisms or have a noxious odor to repel them. For example, poison ivy and several other urushiol-based plants produce an oil called urushiol, which when it is on the surface of a skin, for example, will create a horrible rash. This is a non-specific but effective defense mechanism. Horseshoe crabs are also a good example of an organism with both non-specific immune responses. First of all, they have a shell. Second of all, horseshoe crab have a endolymph, circulatory fluid, which appears to contain proteins which are an absolute defense against bacterial infection. Serum from horseshoe crabs is still used today to detect bacterial contamination in medical supplies because of its apparent infallibility. Given hundreds of millions of years to evolve, horseshoe crabs seem to have nailed it. However, bacteria are only the tip of the iceberg when it comes to invading pathogens. Even bacteria themselves, single-celled organisms, frequently possess multiple defense strategies. CRISPR, which is the new form of genetic editing, which is quite popular and very likely to show up on the test, is an example, as it was originally found forming a viral defense mechanism in simple organisms trying to defend themselves against a specific virus. So CRISPR basically enabled them to recognize when a certain DNA sequence was introduced that was known to be a virus and was able to destroy it. This has since been used by scientists to find specific sequences and cut them out and put them in other places. It's very handy. But it began originally as an immune defense system. Now, another thing you may well be asked to talk about is the immune response of mammals, which is relevant because you and I are probably mammals. We enjoy an active, specific immune response which we are able to develop. It takes many forms in mammals, and it can significantly affect our homeostasis when it is in action. For example, when you are sick, much of the symptoms you experience are due not to the invading organism, but to the effects on your body of your body's immune response by creating circumstances which enable it to more effectively deal with the invader. There are two main types of specific immunity which you should be aware of for the AP Biology test, and in general. The first is commonly called cell-mediated immunity. This makes no sense, because in a way, all things are mediated by cells when it comes to biological processes. But for some reason, this name persists. In cell-mediated immunity, lymphocytes, which is to say white blood cells, of a certain type called T cells, roam around the body, examining the external surfaces of other cells that are part of the organism. When they detect that a cell has been invaded by a virus that has added new concerning changes to its expression of protein, or if they find that a cell is producing incorrect things due to perhaps a malignant cancerous process. In these circumstances, when a cell of the body is found to be treacherous or invaded or mutated, the T cells destroy that cell. This is effective particularly against viral infection and cancerous processes. In a way, you are forming cancers constantly, but your body detects and weeds them out. Successful cancers that come to the attention of clinicians frequently have specific mutations that they have accumulated which enable them to evade this cell-mediated immunity process. And there are drugs which are intended not to kill the cancer directly, but to draw the attention of this cell-mediated immune response to cancers which have previously been ignored. Now, in addition to this cell-mediated immune response, which, if the only thing you remember is T cells, and that they're
killing cells, you'll be in good shape. There is also what is known as the humoral response. This is another terrible name because it makes no sense. However, it is over 100 years old and persists in the literature because of tradition. However, in the humoral immune response, you have another type of lymphocyte, white blood cell, called B cells. The B cells are not directly doing anything. Instead, their job is to circulate in your system and produce antibodies, which are a protein which the B cells make it, and then they drop it into the bloodstream, and then it flows all around in the bloodstream, basically. Okay? These antibodies, their job is to identify pathogens or antigens. The language you need to recognize here is that an antibody is intended to bind to an antigen. And an antigen can essentially be anything that has an external sequence of molecules that forms a shape that an antibody can happen to connect to. It's very much a lock and key process. The best way to think about these antibodies is that they are like wanted posters for criminals. As the, let's say there is let us make the analogy that there is a bacteria that has invaded your body, and that is similar to a criminal that has invaded a town and has begun robbing stores. Okay? When the initial description circulates, there's probably one guy talking to a sketch artist. He can tell you very little about the person who did it. The initial description is very poor, and none of the wanted posters particularly resemble the criminal who is actually being sought. However, as the criminal conducts more crimes and more witnesses are found to describe the criminal, the wanted posters become more accurate, and it becomes harder for the criminal to move around the town without being identified, if that makes sense. Because the B cells are constantly making new forms of antibodies in order to more accurately attach themselves to the invading particles. Now, the wonderful thing about antibodies is, first of all, the B cells generally remain alive in your body for a long time and will continue to make antibodies to a specific antigen if they know that that is a dangerous antigen that must be prevented. So, for example, when we get childhood diseases or vaccinations for childhood diseases, we are training the B cells in our body to continually make small, extremely accurate antibodies which will immediately identify these pathogens if they enter our system. However, when a new antigen is introduced, it typically takes some time for the B cells to learn through random error, and in a sense they kind of evolve themselves over until they're able to produce something that is effective. It's a very effective process, but it takes some time until they're able to make a perfect antibody, and in the meantime they may make some antibodies to attempt to apprehend an invader which happen to cross-react and attack the wrong thing. Like a wanted poster that happens to resemble a normal citizen rather than the criminal. and therefore you end up with the wrong person in prison. Now, when this occurs, it creates what's known as an autoimmune disease, which is when your immune system effectively attacks your own body. An example which springs to mind is myasthenia gravis, which occurs when the body begins to produce antibodies to the acetylcholine receptors in your muscle cells, which causes all of your muscles to fail to work. Just an example. Now, in other situations, these autoimmune diseases can be equally debilitating. But as long as you remember that the body can make antibodies to anything, you'll be in good shape. The idea here is that the reason the antibodies are so deadly, when they attach themselves to an antigen, they change something in their handle, which indicates they have locked onto the correct target. This causes one of two things to occur. Either a passing white blood cell will immediately consume the antibody along with whatever it's attached to, assuming that it must be bad, let's take it in for questioning. The other thing which can occur is the activation of the complement cascade. This is a series of proteins which circulate throughout the bloodstream without any particular purpose until they encounter an antibody which is activated and locked on to an invading organism. When these proteins encounter such a thing, they assemble themselves around that antibody to form a ring and piston arrangement which will activate itself to punch through the cell membrane of the offending organism. Sufficient of these, when they collect on any cell, will break the integrity of its cell membrane and allow all of the cytoplasm to mix with the outside, effectively killing it. This is an automatic process and is quite effective and fast-acting. It explains why antibodies to diseases which were encountered a long time ago are still so effective in attacking these invading organisms when they reappear a second time.